Ever wondered how scientists identify and quantify specific molecules in complex biological samples? Well, they do it using a set of laboratory methods collectively known as blotting techniques. These techniques are visible heroes of molecular biology and biochemistry, playing a critical role in helping researchers study and characterize biological molecules like DNA, RNA, and proteins. Blotting techniques essentially involve transferring these molecules from a gel, where they've been separated based on their size, onto a solid support. This solid support is then used for further analysis, giving researchers a closer look at the molecules they're studying. It's like taking a detailed snapshot of a bustling city from a satellite and then zooming in on each neighborhood for a closer inspection. There are three main types of blotting techniques that scientists use: southern blotting, northern blotting, and western blotting. Each of these techniques has its own specific purpose and procedure, much like different types of cameras used by photographers to capture different kinds of images. Southern blotting, named after its inventor Edwin Southern, is used to detect specific DNA sequences in a sample. Imagine trying to find a specific house in a large city. Southern blotting would be your go-to GPS. On the other hand, northern blotting, a nod to its southern counterpart, is employed to study gene expression by detecting specific RNA sequences. Think of it as trying to understand the demographics of a city, who lives where and in what numbers. Last, but certainly not least, we have western blotting, which is employed to detect specific proteins in a sample. This would be akin to identifying the different types of businesses or services in a city, from restaurants and shops to hospitals and schools. Each of these techniques plays a crucial role in advancing our understanding of biological systems. They are the unsung heroes, the behind-the-scenes workers that allow scientists to unravel the mysteries of life one molecule at a time. So are you ready to delve deeper into each of these techniques to understand how they work? Let's get started. First up, let's explore southern blotting, a technique primarily used to detect specific DNA sequences. The process begins with gel electrophoresis. Here, DNA molecules are separated according to their size. The gel used in this process is typically made of agarose or polyacrylamide. It's a bit like a microscopic obstacle course, with smaller molecules moving faster and therefore migrating further through the gel. Once the DNA is separated, we move on to the denaturation stage. At this point, the DNA within the gel is denatured, breaking the double-stranded DNA into single strands. It's akin to unzipping a zipper, with each tooth representing a base pair in the DNA molecule. Next, we have the transfer stage. The single-stranded DNA is transferred from the gel onto a solid support. This is usually a nitrocellulose or nylon membrane. This step is crucial because it allows us to take a closer look at the DNA, which remains immobilized on the membrane. Following the transfer, we move on to the hybridization stage. The membrane is exposed to a labeled DNA probe that is designed to be complementary to the target DNA sequence. The probe is like a lock, and it's looking for the right key, the target DNA sequence. When it finds it, it binds specifically to it. Finally, we reach the detection stage. The presence of the labeled probe is detected using techniques such as autoradiography. This step is like the grand reveal in a magic trick, where the magician reveals the chosen card. In this case, the magician is the scientist, and the chosen card is the target DNA sequence. So in essence, southern blotting is a 5-step process. Gel electrophoresis separates the DNA, denaturation breaks it down into single strands, transfer moves the DNA onto a solid support, hybridization finds the target sequence, and detection reveals its presence. So that's how southern blotting aids in the detection of specific DNA sequences. Next up, let's discuss northern blotting. Now onto northern blotting, a technique employed to study gene expression by detecting specific RNA sequences. The first step in northern blotting is RNA separation. In this process, RNA molecules are separated based on their size, using a technique known as gel electrophoresis. Typically, scientists use agarose or denaturing polyacrylamide gels. The RNA molecules move through the gel, with smaller molecules traveling further than larger ones, creating a pattern of bands that represents different RNA species. After the RNA has been separated, it's time for the transfer. The separated RNA is moved from the gel onto a solid support. This support is usually a nitrocellulose or nylon membrane. materials that help retain the pattern of the RNA from the gel. Next up is fixation. The RNA is immobilized on the membrane to ensure it stays in place for the following steps. This is often achieved by UV cross-linking or baking, which attach the RNA firmly to the membrane. The fourth step is hybridization. Here, a labeled RNA or DNA probe is introduced. This probe is designed to be complementary to the target RNA sequence, allowing it to bind or hybridize specifically with that sequence. This labeled probe can be detected, making it possible to identify the presence and location of the target RNA on the membrane. The final step in northern blotting is detection. The presence of the hybridized probe is detected using autoradiography or other detection methods. This usually involves exposing the membrane to a film or other detection device. The labeled probes on the membrane give off signals that can be captured and visualized, providing a snapshot of the RNA that was originally in the sample. And there you have it. That is northern blotting for you, a powerful tool to study gene expression. It allows scientists to not only see which genes are being expressed, but also gives them an idea of how much they're being expressed. Now let's move on to Western blotting. Finally, we have Western blotting, a technique used to detect specific proteins in a sample. This powerful tool is widely used in molecular biology, research, diagnostics, and even forensic analysis. Let's get right into the heart of the matter. The first step in Western blotting is protein separation. Proteins are separated based on their size using a technique called polyacrylamide gel electrophoresis, or SDS-PAGE for short. Think of this process like a race where proteins are the runners and their speed depends on their size. The smaller ones move faster while the larger ones lag behind. Once the race is over, we move to the transfer step. Here, the separated proteins are carefully transferred from the gel onto a solid support, typically a nitrocellulose or polyvinylidene difluoride membrane. You can imagine this step as relocating the racers from the track to a podium for further inspection. Next, we have the blocking step. The membrane is treated with a blocking agent to prevent non-specific binding of antibodies. This is akin to setting up barriers around our racers, the proteins, to ensure only the right visitors, the antibodies, get to interact with them. Now we're ready for the incubation with primary antibody. The membrane is exposed to a primary antibody that specifically binds to the target protein. It's like inviting a friend who can recognize a specific racer amongst all. After the primary antibody has found its friend, the target protein, we move to the incubation with secondary antibody. The membrane is then exposed to a secondary antibody that binds to the primary antibody. This secondary antibody is conjugated to a detection molecule, such as an enzyme or fluorophore. It's like adding a spotlight to the friend so they can clearly point out where the racer is. Lastly, we arrive at the detection step. The presence of the target protein is detected through the activity of the detection molecule, often visualized by chemiluminescence or other detection methods. This is the grand reveal, the moment we've been waiting for when we finally see our target protein illuminated and confirmed. And that's how Western blotting helps in detecting specific proteins. So we have explored the three primary blotting techniques: Southern, Northern, and Western blotting. These techniques, each with their unique purpose and procedure, serve as crucial pillars in the fields of molecular biology and biochemistry. Southern blotting, as we've discussed, is used to detect specific DNA sequences in a sample. This is achieved through a series of steps, including gel electrophoresis, denaturation, transfer, hybridization, and detection. The result? A clear, specific visualization of our targeted DNA sequence. Then we moved on to northern blotting. This method is employed to study gene expression by detecting specific RNA sequences. Similar to southern blotting, it involves the separation of RNA molecules, their transfer to a solid support, and the detection of the hybridized probe. This technique allows us to look into the very workings of our genes, their expressions, and their roles in our biology. Finally, we delved into Western blotting, a technique used to detect specific proteins in a sample. This procedure involves protein separation, transfer, blocking, incubation with primary and secondary antibodies, and finally, detection. Through Western blotting, we can specifically identify and quantify proteins, an essential aspect in understanding various biological processes and disease states. These blotting techniques are far from being confined to the realms of academia. They have widespread applications in many areas of life. In research, these techniques are indispensable for studying genetic disorders, infectious diseases, and cancer, among others. In diagnostics, they're used for the detection of specific diseases, providing vital insights for treatment plans. And in the field of forensic analysis, they serve as a means to conduct DNA profiling, helping solve criminal cases and answer paternity questions. Blotting techniques indeed are powerful tools that allow scientists to identify and quantify specific molecules in complex biological samples. They are the unsung heroes of molecular biology, quietly making significant contributions to our understanding of life's complex machinery. Until next time, keep exploring and keep learning.